unfold.
Welcome to the Deep Dive, the place where we take the most pressing questions you have, gather the best available research, and deliver the knowledge you need quickly and thoroughly.
Today, we're tackling something that really gets to the heart of things.
It really does. It's a listener query that's equal parts thrilling and, frankly, deeply unsettling. The core question is this: are you just a taxi for bacteria?
It's a phenomenal starting point because taxi suggests this very simple passive relationship, The host drives and the bacteria are just passengers along for the ride.
Right, you're in control of the vehicle.
Exactly. But the sources we've gathered from metagenomics, neuroimmunology, all over, they suggest that taxi massively, and I mean massively, understates the real dynamic.
It understates the integration, the manipulation, even the control. When you're a taxi driver, you decide the route. You maintain the engine, you can eject a passenger.
Right. You have the agency.
But what we're exploring today is this uncomfortable possibility that the human host may have lost that control, or maybe never really had it to begin with.
Which means we have to move beyond the idea of a single human organism. We need to introduce the central thesis right away. We are not solitary drivers.
Or something else.
We are the Holobiont, a complex interspecies consortium. The host and its trillions of microbial passengers functioning as one interdependent biological unit.
And our mission for this deep dive, then, is to deconstruct how that control actually happens. We need to look at the hardware and the software.
That's it, exactly. Our mission is to analyze the gut-brain axis, the GBA, to reveal the neural wiring, the chemical toolkit, and the evolutionary history that makes this loss of free will. Well, not just a philosophical game, but a plausible, scientifically grounded reality.
We want to show you exactly how the passengers might be taking a turn at the steering wheel.
So to set the stage, let's start by debunking a really famous biological myth, the 10-to-1 ratio.
Oh yeah, everyone's heard this one. Bacterial cells outnumber human cells 10 to 1. It's that classic statistic that makes you feel like a minority shareholder in your own body.
And it's one of the most persistent scientific myths of the last 50 years. It goes back to a rough estimate from Thomas Luckey in 1972. It was really just a back-of-the-envelope calculation.
So not very rigorous. What does the modern, more accurate census tell us?
Well, the numbers definitely shifted, but the horror doesn't really go away. The rigorous modern revision from Sender and his colleagues in 2016, it completely rewrote the numbers.
Oh, so?
They found the ratio is actually much closer to 1.3 to 1.
1.3 to 1. That's a huge change from 10 to 1.
It's a massive shift. For a standard reference man, you're looking at roughly 3.8 trillion bacterial cells versus 3 trillion human cells. So numerically, we're much closer to being on equal footing.
Okay, so if the cell count is almost even, why are we still talking about a massive control mechanism? If we're not so overwhelmingly outnumbered, where does that power imbalance come from?
And this is the absolute key insight. The real measure of microbial power isn't about the quantity of cells. It's about genetic potential.
The genetic operating system.
Exactly. Let's do that comparison. What are we as humans working with?
Our genome, right?
The human genome. It has about 23,000 functional genes. That is our entire toolkit, our complete library of instructions for building and running a human.
And the microbiome.
When you look at the collective genome of our passengers, the hologenome, the numbers are just staggering.
How high are we talking?
We're talking an estimated 3.3 million to 9 million unique genes.
Wait, hold on. 3 to 9 million compared to our 23,000.
That's the comparison. If you run the ratio, you're looking at a disparity of 150 to 1 on the low end and almost 400 to 1 on the high end.
So it's not a numbers game of cells. It's a numbers game of function.
Precisely. We are operating with a vastly smaller instruction manual. Our 23,000 gene human self is fundamentally outgunned in terms of chemical, metabolic, and signaling potential. That genetic disparity is the crux of the whole thing.
Which leads us straight into part 1, the biological redefinition of self, the Holobiont paradigm.
So with a genetic operating system 150 times larger than ours, what can they actually do?
What are the practical implications of that superior chemical toolkit?
They can perform biochemical feats our genome just can't handle. It's like outsourcing critical services. They synthesize bioactive molecules, vitamins we can't make, enzymes we need to survive.
Like digesting fiber, right? That's the classic example.
That's the most famous one. We lack the enzymes to break down complex carbs. Our microbiome takes that material, ferments it into energy for us, and, critically, into signaling molecules.
So they're not just digesting for us, they're communicating.
They're communicating, they're modulating our immune response, regulating inflammation, and, as we'll see, generating neuroactive compounds. The host is just completely outmatched in this chemical arms race.
So we need them for survival. And that means this relationship, this integration, has been cemented by evolution.
This is where the work on the hologenome theory of evolution becomes so central. The argument is that traditional evolution, which focuses on just the host, is incomplete.
So the real unit of selection isn't the human.
It's the holobiont, the host plus all its microbiota, and its collective gene pool, the hologenome. That's what natural selection actually acts on.
So evolution isn't selecting for the fittest human, but for the fittest human microbe community.
Exactly. Human fitness is inextricably linked to microbial fitness. If a certain microbial community is better at extracting nutrients or fighting off pathogens or even inducing behaviors that help the host get resources,
Then the whole unit, the whole holobiont, is selected for survival.
yes, and this is where the sinister idea of manipulation really gains its evolutionary footing.
Right, because if a microbe's main goal is to replicate and spread, and it needs us to do something specific to achieve that, like eat a certain food or be more social so it can be transmitted.
Then evolution will favor the microbes that are best at making the host perform that behavior. We're not just a vehicle, we become an extended phenotype of our microbial passengers.
Which brings us to the psychological part, which is maybe the scariest bit. The illusion of autonomy. When I feel a strong emotion, like a surge of panic or a craving I can't resist, my conscious mind just makes up a story to explain it.
You rationalize it. I am hungry, I am anxious.
I call it my will.
But the holobiont view forces us to see those stories as post hoc rationalizations, after-the-fact excuses. If your microbial passengers can flood your system with neurochemicals, changing your baseline levels of serotonin or dopamine or GABA,
They're setting the mood music.
that's a perfect way to put it. They're curating the playlist, and the conscious mind is just trying to dance to it, inventing reasons for the moves after they happen. The self is a negotiated state.
But surely there's a difference between wanting a chocolate bar and a huge life decision, like changing careers or choosing a partner.
It might be a difference of degree, not of kind. Think about it. If your microbial community can profoundly shift your baseline anxiety, making you less risk-averse or induce chronic low-level depression, making you less ambitious.
Then those shifts affect the emotional filter for all of your decisions.
Exactly. Your major life choices are often just rational narratives built on a microbial-influenced emotional foundation.
That's a fundamental challenge to the idea of agency. So if they're in control, they must have a communication network. Let's get into the mechanics. The superhighway. Part 2. The neural hardware. The gut-brain axis.
The GBA. This is the infrastructure that lets chemical demands become neural commands. It's a sophisticated bidirectional system connecting the gut to the central nervous system.
And the main physical bridge, the sort of fiber-optic cable of this whole system, is the vagus nerve.
Cranial nerve X. It's called the wandering nerve for a reason. It runs from the brainstem all the way down, innervating most of the major organs, including the colon. It's the primary high-speed connection.
But the key vulnerability, the thing that really gives the gut this power over the brain, is the asymmetry of the nerve itself.
This is absolutely critical. Studies show that 80 to 90% of all the fibers in the vagus nerve are afferent.
Meaning they carry signals from the gut up to the brain.
Exactly. Only 10 to 20% of the traffic goes the other way, from the brain down to the gut.
That completely flips the power dynamic. It means the brain is overwhelmingly a receiver, not a commander. The gut is constantly updating the brain, but the brain has very limited bandwidth to send commands back.
The tail is wagging the dog, structurally speaking. The brain is basically eavesdropping on the chemical conversation happening in the gut.
So how does a chemical message from a bacterium actually get turned into a neural signal that travels up that cable?
It starts at the gut lining. There are these specialized sensory cells called enterochromaffin cells, or EECs.
And these cells are like the taste buds of the gut.
They literally taste what's in there.
Yeah.
They have receptors for bacterial components like bits of cell wall called LPS, and most importantly, those key bacterial metabolites, the short-chain fatty acids, or SCFAs.
Okay, so the EECs are the first responders. They taste a microbial signal. What happens next?
They immediately dump huge quantities of neurotransmitters. The main one is serotonin, 5-HT, and this is a point that you just can't overstate. 90 to 95% of your body's entire supply of serotonin is produced and stored in the gut.
Which is incredible. The brain uses serotonin for mood, sleep, appetite. But almost all of it originates right there where the microbes are.
Yes. So this serotonin gets released by the EECs, and it binds to receptors on those upward-bound afferent terminals of the vagus nerve. That binding instantly converts the chemical signal into a high-speed electrical impulse.
And it shoots straight to the brainstem. Where does it land?
It enters at a place called the nucleus tractus solitarius, or NTS. The NTS is the main visceral processing center. It's like a switchboard. It decides where to relay this urgent message from the gut
And where does it send it?
to several critical higher brain centers that govern our baseline emotional state. It hits the locus coeruleus, which regulates arousal and stress. It hits the dorsal raphe nucleus, a major hub for mood and anxiety. And critically, it hits the amygdala.
The amygdala, the brain's fear and threat detection center.
Precisely. This is what we call bottom-up modulation. So imagine a sudden bloom of a specific bacteria in your colon. This triggers a massive serotonin spike from your EECs. That signal flies up the vagus nerve, hits the NTS, and activates your amygdala.
And the host, you suddenly feel this overwhelming anxiety or fear. But there's no external reason for it. The panic feels internal because the threat is internal.
And it all happens in a fraction of a second, completely bypassing your rational, conscious prefrontal cortex. Your thinking brain is the last to know, and its job is just to invent a plausible story for the terror your body is already feeling.
That's incredible. Now, beyond the vagus nerve, there's another piece of hardware. The enteric nervous system, the ENS, the second brain.
The ENS is massive. It's a dense network of 100 to 500 million neurons embedded right in the walls of the gut. That's as many neurons as you have in your entire spinal cord.
It runs things locally, but it also interfaces directly with the microbes.
Exactly. And we know microbes are essential for its development. Germ-free mice with no microbiota have significant structural and functional problems in their ENS. The microbes are co-architects of our own hardware.
And because it's a neural network, it's also a huge neurotransmitter warehouse.
A huge one. Serotonin, dopamine, GABA. The ENS produces all of them, basically modulating the volume and the tone of the messages being sent up to the main brain.
Okay, so the hardware is in place. But what about the final fortress? The blood-brain barrier, the BBB. Surely that keeps the brain safe.
It's designed to, yes, it's a very selective filter, but it is not a perfect wall. Microbes have evolved both indirect and direct ways to get messages or even themselves across.
Let's start with the chemical infiltration.
This involves those tiny bacterial products we mentioned, the SCFAs, butyrate, propionate, acetate. They're so small and lipid-soluble they can get into the bloodstream and just cross the BBB. And once they're in the CNS, they start influencing things. They interact directly with brain cells, especially microglia, the brain's immune cells. They can modulate inflammation, they can alter neurotransmitter release.
And the microbes can also just weaken the barrier itself, right? Make it leaky.
That's the second strategy. The gut microbiota actually regulates the tightness of the BBB. If you have dysbiosis, an imbalance, it can compromise the barrier's integrity. That lets larger neurotoxic molecules like LPS from bacterial cell walls slip through.
And LPS in the brain means neuroinflammation.
Precisely. It's linked to depression, Alzheimer's, Parkinson's. The microbes can indirectly poison the brain just by making the gatekeeper weak.
And finally, the most direct method, the Trojan horse.
This is what evolved pathogens like Toxoplasma gondii do. They have the ability to hijack the host's own immune cells, like macrophages. The parasite hides inside the cell.
So the cell that's supposed to be defending the brain is actually carrying the invader across the barrier.
It's the ultimate deception. The immune cell follows its normal chemical cues, crosses the BBB, and delivers the parasite directly into the brain tissue.
Okay, so the hardware is there. The fortress can be breached. Now let's get into part 3, chemical warfare. The actual software of control.
This is where we name names. That huge gene library we talked about isn't just sitting there. It's a factory churning out psychoactive compounds. We can map specific bacteria to key parts of our psychology.
Let's start with the one that calms us down, GABA.
GABA is the brain's main inhibitory neurotransmitter. It's what puts the brakes on anxiety. And genera like Lactobacillus and Bifidobacterium are prolific GABA producers.
And there's a great example of this, isn't there?
A perfect one, Lactobacillus rhamnosus JB1. In animal models, it alters the expression of GABA receptors in the brain, and it does it via the vagus nerve. It basically presses the host's chemical calm down button.
That is direct control over our emotional state. What about the other way? The reward and motivation systems driven by dopamine.
Genera like Bacillus and Serratia produce dopamine, contributing to our baseline reward signaling. But as we'll see later, the most explicit hacking of this system comes from organisms that can just make dopamine themselves.
And we already know serotonin is largely driven by the gut.
Right, and specific genera like Candida and Streptococcus are directly involved. Same for norepinephrine, the stress and arousal chemical produced by Escherichia and Proteus. The microbes have a full chemical control panel for our emotions.
This dependency creates a massive vulnerability, what the sources call the tryptophan trap.
This is one of the clearest biochemical links between gut health and mood disorders. Tryptophan is the essential amino acid we need to make serotonin.
We have to get it from our diet.
We do. And under conditions of inflammation or stress, certain bacteria can activate a host enzyme called IDO.
And what does IDO do?
IDO is the enzyme that enforces a chemical detour. It shunts all the available tryptophan away from the serotonin pathway and pushes it down the kynurenine pathway.
So it's a microbial roadblock. The brain is starved of the precursor it needs to make its primary mood regulator.
Instantly, leading to a direct depletion of central serotonin, which is the biochemical definition of clinical depression. But it's worse than that. The kynurenine pathway also produces neurotoxic byproducts.
Like what?
The most notable one is quinolinic acid. It's toxic to neurons. So when the trap is sprung, you are simultaneously depressed from serotonin starvation and being poisoned by neurotoxins.
Which perfectly explains that profound lethargy, that social withdrawal of sickness behavior.
It's a debilitating manipulation. A targeted microbial weapon could induce a state of profound, persistent depression just by starving the brain.
So moving from neurochemistry to genetics, let's go back to the short-chain fatty acids. We know they cross the BBB, but what they do there is described as almost science fiction. They are epigenetic editors.
This is maybe the deepest level of control. SCFAs, especially butyrate, don't just influence existing neurochemistry, they fundamentally alter the expression of our host genes.
How does a tiny byproduct of fiber fermentation do that?
Butyrate acts as a powerful histone decetylase inhibitor, or HDAC inhibitor. Your DNA is coiled tightly around proteins called histones. HDACs are the enzymes that keep the DNA coiled up tight, silencing the genes in that region.
So by inhibiting the silencers, butyrate.
It forces the DNA to relax. It keeps the chromatin open. And if the chromatin is open, the host cell can read those genes and express them.
So tiny bacteria, just by eating fiber in my gut, can produce a metabolite that gets to my brain and literally unlocks sections of my genetic operating system.
That's the staggering implication. They can alter the long-term gene expression profile of your brain cells, influencing neuroplasticity, memory, mood. They aren't just influencing you today, they're subtly rewriting the instruction manual for your future self.
And we see clear behavioral links to this.
Absolutely. High levels of propionate have been linked to autism-like behaviors in rodents. Butyrate, on the other hand, is generally neuroprotective and acts as an antidepressant. The very architecture of our mind is being tuned by these microscopic byproducts.
Now we get to maybe the most insidious strategy, hormonal hijacking and molecular mimicry. This is where the passengers forge host identity cards.
This strategy allows microbes to either directly activate our receptors with fake keys or, even more cleverly, trigger an autoimmune response that disrupts our natural balance. Let's focus on the satiety signal.
Alpha MSH, the hormone that says I am full.
Correct. It binds to the MC4R receptor in the hypothalamus and puts the brake on hunger. But common gut bacteria like E. coli produce a protein called CLPB.
And CLPB looks suspiciously like our satiety hormone.
It shares a critical amino acid sequence. It's a molecular mimic.
Yeah.
And it has two ways of interfering.
What's the first, more direct way?
It can sometimes bind directly to our MC4R receptor, creating a false temporary feeling of being full.
But the more dangerous long-term attack uses our own immune system against us.
Exactly. The immune system sees this foreign bacterial protein, CLPB, and creates powerful anti-CLPB antibodies to attack it. But because of the mimicry, those antibodies are cross-reactive.
So they don't just attack the bacterial protein.
They attack the host's own alpha MSH. Our own immune system starts neutralizing our natural I am full signal.
The brake on appetite is removed,
leading to persistent, unregulated hunger. And this isn't just a theory. High levels of these cross-reactive antibodies are found in patients with severe eating disorders like anorexia, bulimia, and binge eating disorder.
So these complex psychological conditions could have their roots in a microbial identity theft that triggers a destructive autoimmune response.
The evidence strongly points that way. And the mimicry extends to other hormones too, like ghrelin, the hunger hormone, and leptin, the satiety hormone. Microbes can make fake versions of those too.
Which brings us to the craving control loop. The idea that our cravings aren't really ours, but are intense demands from specific microbial guilds guarding their resources.
We can link specific cravings to specific species.
Uh-huh.
Take Prevotella copri. It thrives on complex carbs and fiber. When Prevotella is dominant, the host has overwhelming carb cravings.
And it's not just a negative feeling if you don't comply. It actually hacks the reward system to make compliance intensely pleasurable.
It's so sophisticated. High Prevotella abundance actually modulates the dopamine transporter in the brain, intensifying the dopamine reward you get from eating its preferred food. You get an outsized pleasure hit for feeding the correct passenger.
It's behavioral conditioning from the inside out. Now, what about the flip side, the candida sugar trap? This sounds like an addiction cycle.
That's exactly what it is. Candida, a yeast, thrives on simple sugars. When it ferments sugar, it produces alcohol and the neurotoxin acetaldehyde.
Which is what makes hangovers so miserable.
Right. And in the gut, it causes brain fog, irritability, fatigue, the classic sugar crash. The yeast creates a cycle. When your blood sugar drops, the yeast's waste products make you feel severely ill.
And the only way to stop the misery
Is to consume more sugar. It alleviates the crash symptoms, silencing the yeast's distress signals. It's a dependency loop that functions identically to addiction. You're eating sugar not for pleasure, but to pay a chemical debt and stop the suffering caused by your passenger's withdrawal.
My sweet tooth might just be a hostage negotiation.
A debt you have to pay multiple times a day.
To really drive this home, we have to look at nature's most extreme examples. This is part 4, Puppet Masters of the Wild, Parasitic Precedents.
We have to start with Toxoplasma gondii. This parasite infects about 1/3 of the global human population. Its life cycle requires it to get from an intermediate host, like a rat or a human, into its final host, a cat.
And to do that, it has to completely override the rodent's most basic survival instinct.
It engineers what's called the fatal feline attraction. The infected rodent, which should be terrified of cat urine, suddenly becomes attracted to it. It seeks out the cat, gets eaten, and completes the cycle.
And we know the mechanism for this is its dopamine hack.
Right. The parasite itself has 2 genes for tyrosine hydroxylase, the key enzyme for making dopamine. It can synthesize dopamine directly inside cysts in the host's brain.
Flooding the reward centers and overriding the fear response. And the human effects are just as chilling.
Infected humans show a pattern of increased risk-taking and a suppressed fear of failure. They're 2.65 times more likely to be in a traffic accident.
And it's even linked to entrepreneurship.
The boldness, the reduced risk aversion, it correlates with a higher likelihood of starting your own business. And on the darker side, it's linked to schizophrenia, intermittent explosive disorder, uncontrollable rage, and higher suicide rates.
Okay, another master manipulator, the rabies virus.
Rabies is all about transmission efficiency. It enters the peripheral nervous system and immediately hijacks the neuron's internal machinery to get to the brain as fast as possible.
It boards the express train to the CNS.
It does. And once it's in the limbic system, it engineers 2 key behaviors. First, aggression to provoke biting, and second, the bizarre but critical symptom of hydrophobia.
The fear of water. Why is that necessary for the virus?
It causes painful spasms in the throat if the host tries to swallow. This ensures the virus-packed saliva isn't washed away. It concentrates the vector in the mouth, maximizing the viral load for the next bite. It's ruthlessly efficient.
And finally, the gold standard of somatic control, where the brain isn't even the target. The zombie ant fungus, 𝐏𝐿𝐴.
This is the definitive proof that you can lose autonomy even if your consciousness is intact. The fungus infects a carpenter ant, and crucially, studies show it does not invade the brain.
So it bypasses the central processor and goes straight for the motor controls.
It colonizes the muscles, forming a dense network around the muscle fibers. The brain might still be sending signals, but the fungus has overridden the commands.
What's the final command it issues?
At solar noon, when conditions are perfect for the fungus, it releases metabolites that cause the ant's jaw muscles to hypercontract. It makes the ant bite down on a leaf vein and never let go.
A permanent, involuntary death grip.
The ant is a prisoner in its own body. It's the ultimate proof that a parasite can separate will from action.
Which leads us to our final section, Part V, the transferable self. If mood and personality are driven by microbes, then swapping the microbes should swap the personality.
And this is exactly what fecal microbiota transplantation, or FMT, studies have proven. It's the most direct, compelling, and frankly, terrifying evidence we have.
Let's start with the depression transfer study from 2016.
Researchers took fecal samples from human patients with major depressive disorder and gave them to rats that had their own microbiota wiped out.
And the rats became depressed.
They developed classic depressive behaviors: anhedonia, where they lost interest in pleasure, anxiety, inflammation, and that altered tryptophan metabolism. The tryptophan trap was successfully transferred.
So depression is, at least in part, a transmissible dysbiosis.
And the same applies to personality. Look at the Bursic switch study. They used 2 strains of mice, one naturally timid and anxious, the other naturally bold and explorative.
And they just swapped their gut microbes.
They swapped them. The timid mice became bold, the bold mice became timid and anxious.
So fundamental personality traits like boldness and shyness are largely determined by our microbial passengers.
It suggests that a huge part of what you think of as your inherent temperament is actually a microbial phenotype.
And this even works across age. The 2024 study on rejuvenating the mind.
FMT from young, sharp mice into old mice with cognitive decline actually reversed that decline. It restored hippocampal plasticity and memory, largely driven by beneficial SCFAs from the young donor.
This all points to a strange future, doesn't it? The search for premium stool as a biological commodity.
Which brings us to the double-edged sword of psychobiotics. If we can use specific bacteria to heal the mind, we can also use them to control it.
So psychobiotics are live organisms that provide a mental health benefit. What are some of the therapeutic strains?
Take Bifidobacterium longum 1714. It's been shown to reduce cortisol and anxiety in stressed humans by blunting the body's central stress response. It literally dials down panic.
And Lactobacillus rhamnosus JB1, the one who works on GABA.
Yes, and crucially, they proved its effect depends on the vagus nerve. If they cut the nerve, the anxiety-reducing effect disappeared completely.
That is irrefutable proof of the neural superhighway in action.
And that specificity is why the potential for weaponization is so high. If one strain can induce calm, a modified strain could be engineered to induce apathy or fanaticism or targeted aggression.
You could create a compliance strain, introduce it into a food or water supply, and render a population docile.
Or the opposite, an aggression strain that targets the amygdala, creating a state of hypervigilance and rage optimized for conflict. The line between therapy and control is microscopically thin.
So let's synthesize all of this. Let's summarize the architecture of control into 3 tiers of manipulation.
It moves from subtle conditioning to outright physical override.
Let's start with Tier 1, metabolic conditioning.
This is the carrot and stick approach. Agents like Prevotella and Candida. The mechanism is simple: reward with dopamine for compliance, punish with toxins and anxiety for resistance. The manifestation is your cravings, that hangry feeling.
Tier 2, neuroendocrine hacking.
This is identity theft. Agents like E. coli, Toxoplasma. The mechanism is molecular mimicry, direct neurotransmitter synthesis, and epigenetic editing. The manifestation is personality shifts, mood disorders, eating disorders.
And finally, Tier 3, somatic override.
This is the zombie protocol, the extreme examples like Ophiocordyceps and rabies. The mechanism is direct invasion and hijacking of the neuromuscular junctions. The manifestation is a catastrophic loss of control. Violent aggression, permanent incapacitation.
So let's bring it back to the beginning. We asked if we were just a taxi for bacteria. After all this, the answer is so much more disturbing than a simple taxi ride.
The evidence confirms that the self is, in many ways, a biological fiction, a negotiated reality. Your cravings are bacterial text messages. Your anxiety is a microbial weather report. Your personality is a microbial phenotype.
So we're not the driver. It seems we're the entire system. The car, the roads, the fuel, and the passengers have learned exactly which wires to cross to make us drive where they need to go.
We are the Holobiont. And what this all suggests is that the free will we hold so sacred is maybe just the veto power of the prefrontal cortex. A veto that, as we've seen, can be chemically overridden.
So here's the final provocative thought for you to explore, based entirely on the source material we've just dissected. If fecal microbiota transplantation can transfer major depression and switch fundamental personality traits like boldness and timidity, then how much of your next major life decision, your career change, the choice of your romantic partner, or your biggest financial gamble was fundamentally influenced not by your rational self, but by the trillions of tiny passengers vying for resources and replication? And what does that mean for your destiny?
Something to mull over. We hope this deep dive gave you much to consider.
Thank you for joining us. We'll see you next time.